common to mammalian cell culture, they also affect bacterial culture. Indeed, one of the many major contributions of Bruce Ames was to develop the Ames test. If you do the Ames test on various bacteria, you often find lots of things to be mutagenic or to be classified as mutagenic, but in fact, we probably are not mutagenic. Mutagenicity of tea. Now, maybe if you don't like tea, you can drink coffee. Well, maybe not, because here's another paper showing that coffee is highly mutagenic. Now, as far as we know, studies on humans have shown that coffee and tea are probably good for you, or at least may not do you any harm, unless you start drinking ridiculous amounts. So what is the origin of this artifactual mutagenicity in Salmonella in the Ames test? And again, it's simply hydrogen peroxide. Commonly drunk beverages like instant coffee, black tea, brown coffee, even green tea, contain hydrogen peroxide. And so instant coffee, if I make it and I leave it sitting on the bench, I'm not talking about a little bit, I'm talking about a few hundred micromolar. And of course, when you drink your tea or coffee, basically this hydrogen peroxide diffuses into the buccal or esophageal epithelium and is quite rapidly disposed of. Funnily enough, when you add milk, and this is adding 10% milk to the beverages I showed you earlier, you cut the content of hydrogen peroxide by about half, because milk contains various constituents that can scavenge hydrogen peroxide. So if you look at the instant coffee in this study, it's 250, putting in 10% milk cuts it to about 150. Similarly for green tea. Now, nobody would ever add milk to green tea. This is just a laboratory experiment. So milk scavenges hydrogen peroxide. Now, the other reason, apart from hyperoxia, is that cell culture is an oxidative stress, is that cell culture media are often lacking the key diet-derived antioxidants. Papers from Regalius Flohey and their colleagues, Gary Wetner, show that in fact many cell culture media are somewhat deficient in selenium, and selenium is a cofactor of thyrodoxine reductases and certain glutathione peroxidase. So if you don't have enough selenium, you can't make these defenses properly. There's often not much vitamin E in cell culture because there's not much lipid. There's obviously some when you add fecal calf serum or its equivalent. There's often not a lot of vitamin E. And very few widely used cell culture media contain vitamin C. And this is a very important lack, because vitamin C, as I mentioned earlier, is a cofactor for a number of enzymes. And I'll come back to that in a moment. Now, one of the many great achievements of Linus Pauling was to raise the concept that high-dose vitamin C could be useful in the treatment of cancer. And this was very popular when he proposed it. Then it went for a period that people didn't believe it. Now there is a resurgence, so we'll see what the clinical data eventually show. And one of the reasons he believed that was a range of studies showing that vitamin C is toxic to cancer cells in culture. And some years ago, we decided to go back and reinvestigate this. And I was working with Marie Claymont and her colleagues at the National University of Singapore. So in this experiment, we're using HL60 cells, which is a leukemic cell line. They're in Delbecco's modified Eagles medium, DME, which doesn't contain vitamin C. They'll start dying. So 500 micromolar, 30–40% are dying. By the time you get to the millimolar range, many of them are dying. Now, in fact, if you go high enough with vitamin C, they all die. But you switch at one stage from more apoptotic cell death, as shown here, to a more necrotic cell death. The other thing that we observed was that if we didn't use DMEM, but another medium, RPMI, the cytotoxic effects of ascorbate almost disappear. Now, if ascorbate is really toxic to cancer cells, what difference should it make what medium you're using? But it clearly does. And in fact, what we discovered, to cut a long story short, was that ascorbate reacts with DMEM and it makes hydrogen raise, the cytotoxicity disappears. So in other words, the apparent cytotoxicity of ascorbate to cells in culture in DMEM is an artifact of cell culture. Now, this was published a while ago, many leading journals like Nature Genetics, where people are looking at the effects of being completely unaware that it can react with cell culture media and make hydrogen peroxide. And indeed, some of its positive effects might be due to low-level hydrogen peroxide generation, as I showed you in the studies of Roy Burden earlier, where a little amount of hydrogen peroxide actually accelerates cell proliferation. The other issue, of course, with ascorbate, as I mentioned, is that it's a cofactor for many enzymes, and especially the TEC dioxygenase enzymes that catalyze the oxidation of 5-methylcytosine in DNA. And this is the first step in DNA demethylation. So if you don't have ascorbate in your cell culture media, your cellular DNA may become hypermethylated, and that might give you all kinds of metabolic effects. And depending on the ascorbate you add and the cell culture medium, you can get various levels of hydrogen peroxide, which can do various things. And having discovered this with ascorbate, we wondered, does it happen with other biomolecules? Yes, it does. It particularly happens with the flavonoids and other polyphenolic compounds. They react very rapidly with cell culture media. They generate hydrogen peroxide. They also generate quinones and semiquinones. And again, many of the reports of the cytotoxicity of polyphenols to cells in culture are at least in part due to hydrogen peroxide generation. So you really need to be aware of this artifact. Now, even with compounds, phenolic compounds that don't generate hydrogen peroxide, such as resveratrol and curcumin, they are unstable in cell culture media. This is simple HPLC studies with those two compounds showing that the levels drop with time and that the effects they have on cells in culture may not only be direct effects, they may be effects of the degradation product. So whenever you add anything to cell culture media, it's worth checking that in fact whether they're reacting with the media or not, and if so, what they're producing. So apart from our studies on hydrogen peroxide, the second major area into which I researched, and a lot of this was with my esteemed colleague John Gutteridge, was the role of iron in promoting oxidative damage. And this is a fairly old table. Of course, the classic example of iron catalysis is its reaction with hydrogen peroxide, producing hydroxyl radical and maybe other reactive oxometal species, its ability to decompose lipid peroxides into peroxyl and alkoxyl radicals, then on to cytotoxic aldehydes, and the ability of iron to promote the oxidation of thiols, of nicotinamide, reduced nicotinamide nucleotides, of vitamin C, and of many neurotransmitters. All these molecules are very autooxidizable, but the autooxidation is greatly accelerated by traces of transition metal ions. And one of the major antioxidant defenses in the human body, we all know about SOD and the peroxyredoxins and glutathione and so on, is that this catalysis by iron involves what's often called catalytic ion complexes, which are Fe2 plus, sometimes Fe3 plus, loosely bound to various biomolecules. And one antioxidant defense is to make sure that the levels of catalytic iron in vivo are as small as possible. And indeed, we know that this is important because in certain diseases, such as idiopathic hemochromatosis, this iron sequestration fails. There are catalytic iron complexes in the plasma and tissues, which at least some of it is iron citrate. And this was an early paper we did with Peter Sadler identifying that. And they catalyze damage. And if you look at the consequences of untreated hemochromatosis, they are devastating: severe joint damage, liver damage, hepatocellular carcinoma, early onset diabetes, and so on. So if you don't sequester iron properly, you have a lot of problems. And one of the things that John and I worked on is the concept that when tissues are damaged, iron is liberated. And in fact, this is especially true of human brain. If I take some human brain material and I homogenize it, iron is very rapidly released and I get rapid liquid oxidation. The same is true of other tissues, but not to the same extent. And working with John and Pat Evans and many others, Adrian Bomford, Okazi Aroma, and so on, we described a number of situations in which you do have catalytic iron, including some premature babies, cancer chemotherapy, because it kills a whole load of cells, liberates iron, you get a transient iron overload, liver failure, when the liver is dying, iron is released from the damaged liver, and so on. And these are all examples of aggravated oxidative damage due to the release of catalytic iron. Another area that we made some contribution to is the role of iron in atherosclerosis. And basically, we showed that advanced atherosclerotic lesions in humans, the so-called gruel within the lesion contains catalytic iron. In order to study this from the earliest stage, we used an animal model, a high cholesterol diet. They developed atherosclerosis. And we like this model because it doesn't involve genetically manipulating ApoE status or anything else. It's just a diet that is very bad for the rabbits. And if you look, you will see that the normal vessel wall contains some iron because, of course, there are cytochromes and other heme proteins and so on. But when you start feeding cholesterol, the blue area is beginning atherosclerosis, and that shows an accumulation of iron. So that in the early atherosclerotic lesions, there is more iron than normal. And if I summarize that, this was 6 different experiments on different groups of rabbits. And what we showed was that the more extensive the atherosclerosis, the higher the content of iron in the atherosclerotic lesion. Now, the techniques we were using to measure iron were with the Department of Physics at the National University of Singapore, and they had the capacity to image a large number of elements. The other one that we noted was zinc, that again, apart from one of our 6 batches of experiments, the atherosclerotic lesions were enriched in iron, but depleted in zinc. Well, of course, these are just observations. Do they mean anything? Well, in fact, we used desferrioxamine, and when you fed the animals desferrioxamine, it inhibited atherosclerosis quite slowly. It took about 9 weeks because desferrioxamine is not very good at depleting lesion iron. And in fact, sometimes in the first couple of weeks, it increased it and then it went down. But when the iron went down, the atherosclerosis became less prominent. And similarly, when we gave our rabbits extra zinc in the diet, or when we bled them by phlebotomy, the lipid peroxidation went down and the development of atherosclerosis went down. So at least in cholesterol-fed rabbits, iron promotes atherosclerosis and zinc inhibits it. The paper on the right of this slide is the original paper of John Gutteridge and Tom Dormandy and their colleagues about the fact that brain, when damaged, readily undergoes lipid peroxidation. And in fact, they were using an assay using brain homogenate to measure inhibitors of lipid peroxidation. Now, among the diseases where we know oxidative damage is important are the neurodegenerative diseases. And if we look at the affected brain regions of all the major neurodegenerative diseases, we see certain key factors. There's something wrong with mitochondrial function. There is an accumulation of abnormal proteins like plaques and tangles in Alzheimer's disease, Lewy bodies, Parkinson's disease, and so on. There is increased oxidative damage, and there is iron deposition. These changes are common to all the diseases, although there are some differences. For example, the major mitochondrial defect in Parkinson's disease is in complex 1, whereas in, for example, dementia, it's more in complex 4. But there's always a common feature. So when I look at and this was work we did with Peter Jenner when I was working at King's College London, and it's also work from many other groups. If we look at the affected brain regions of Parkinsonian patients, these are post-mortem brain samples, you see increased levels of multiple products of lipid peroxidation, of A2-HTG, of A2-HTG. These are oxidative damage products to DNA and RNA, and increased protein carbonyls and other things. And the longer the subject has had Parkinson's disease, the higher these levels go up. Quite interestingly, when we studied live Parkinson's patients, and of course you can't take brain material from them, and we measured biomarkers of oxidative damage in the plasma, they were also elevated, but quite interestingly, and also we measured urinary A2-HTG, but quite interestingly, the levels tended to go down with length of the disease. And whether that's the disease or the treatment, we don't actually know. Even more prominently, when we look at the affected brain regions of subjects who passed away with Alzheimer's disease, we see a very marked increase in biomarkers of oxidative damage to all biomolecules. And some oxidized proteins include copper, zinc, SOD, and also proteins involved in the ubiquitin proteasome cycle, so that when they're oxidized, you can't turn over aggregated proteins properly. And one major pioneer in this work was Alan Butterfield, and I recently had the privilege of summarizing some of this work and generating new ideas in a paper in Nature Reviews Neuroscience. Now, one very important thing that Alan and his colleagues showed was that if you look at subjects, or you look at the brains of subjects who passed away with mild cognitive impairment, the oxidative damage is already there. So it's already elevated in MCI by several biomarkers, which suggests that it may be an early stage in AD development, and therefore it should be amenable to antioxidant therapy. Of course, it doesn't prove that it's a key part, but it's very interesting. It's an early event. So what happens is I take people with these diseases and I start giving them antioxidants. And the ones that have been most studied are vitamin E. And in general, they're not very effective. Occasionally they're deleterious, especially the case of beta-carotene in smokers with lung cancer, which is contraindicated because there's evidence that it accelerates the development of the lung cancer. The one that's been most studied is vitamin E. And early studies in Alzheimer's showed modest protective effects of high dietary doses of vitamin E. Later studies didn't confirm that. It doesn't seem for a number of other diseases. And high dose vitamin E for long periods is not necessarily harmless. There is some suggestion. Now, how important it is not clear. There is some high doses for very long periods will increase your risk of hemorrhagic stroke. So obviously giving high doses of vitamin E doesn't seem to help very much. And large number of explanations have been given for this, but I think we have to face the very important fact as illustrated by this cartoon, but however we argue it, it doesn't work. Well, does this mean antioxidants are going to be completely useless in the treatment of disease? Well, why did many of the trials fail? Well, a lot of them studied advanced disease and not early stage disease, where oxidative damage may be more important. They often used people who already had a good intake of antioxidants. They may have used too high a dose. They may have used the wrong time scale and so on. But certainly for neurodegenerative diseases, when you feed vitamin E, it takes a very long time to raise the brain level. So the brain resists taking in too much vitamin E. So there are all kinds of reasons. Now, the other obvious reason is, of course, that maybe oxidative damage is not important in these diseases. But a very large amount of data suggests that it is important in the development of a number of cancers, and it is important in neurodegenerative disease. It may well be important in cardiovascular disease, although to my mind, it's not as clear-cut. So if oxidative damage to various biomolecules contributes to the development of cancer and to development of neurodegeneration, why doesn't giving high doses of antioxidants actually work? Well, one assumption that was made in these studies was when we give high-dose antioxidants, we decrease oxidative damage. And if we decreased oxidative damage, we should decrease disease severity. But none of these early intervention studies actually measured oxidative damage. So some years ago, we took healthy human subjects, we gave them high doses of vitamin C, beta-carotene, vitamin E, and we measured, as far as we can, biomarkers of lipid peroxidation and DNA damage, which of course you can measure in urine, you can measure in blood. Of course, you can't see what's going on deep within the tissue. But what we found, many other people found the same thing, that in people who have reasonable diets, giving high doses of vitamin E, vitamin C, in some studies carotenoids, in some studies polyphenols, did not decrease oxidative damage. And if it didn't decrease oxidative damage, then it wouldn't have an effect on the disease progression. So this gives you a lot of problems. I've raised a big problem here, but in fact there is light at the end of the tunnel. So I can ask the question, if giving high doses of diet-derived antioxidants to people doesn't decrease oxidative damage, doesn't affect oxidative damage, what does? And this is a summary of work from multiple laboratories, but a number of the factors that we know influence the risk of disease also influence oxidative damage. So obesity causes oxidative damage. So does hyperglycemia and hypercholesterolemia. So if you're an obese diabetic, you are better off losing weight, which is easier said than done, of course, controlling your blood glucose, controlling plasma lipids, and that will bring down your oxidative damage. And it may well be that these effects of lifestyle interventions are mediated by decreasing oxidative damage. There are some things, of course, that increase oxidative damage: cigarette smoking, environmental toxins like arsenic and nitrogen dioxide, excessive exposure to sunlight, and so on. And of course, the other thing we know, as I mentioned earlier, is that diet rich in vegetables are associated with decreased risk of many diseases. Although whether that's the antioxidants or something else or a combination of things, we can't be certain of at the moment. So the major challenges in the ROS field are, of course, we need to develop antioxidants that work, that do decrease oxidative damage, but they don't decrease it so much that they interfere with the normal cellular function of ROS. The role of ROS in aging is completely all over the place, so we need to do it. And of course, when we know the true role of ROS in aging, maybe we can do something about aging. Our knowledge of redox signaling has developed enormously, but it's still very embryonic, and we need to understand it. We need to understand it better. So the fact that antioxidants rarely change oxidative damage, but that things that do decrease disease incidence also decrease oxidative damage should in fact make it effective. There have been a lot of studies on mushrooms recently. Now there are many toxic mushrooms, so here I'm talking about the mushrooms. This was a study that did in Singapore, and it was following on from a very similar study in Japan, which showed the more mushrooms you eat, the less likely you are to have mild cognitive impairment. Other studies have in fact shown that high mushroom consumption is associated with lower risk of cancer. Well, so what? This is just correlation. And in fact, fruits and vegetables generally. But I will come back to mushrooms in a moment. So what antioxidant agents might actually work in vivo? Well, I've emphasized the key role of iron, and therefore a number of groups are studying iron chelators, such as desferrioxamine. Now, desferrioxamine is a complicated molecule. It binds iron in a redox inactive form, but it also activates the HIF-1 alpha system, so it has multiple physiological effects. And a number of groups are studying the role of desferrioxamine in the treatment or prevention of disease. And similarly, other iron chelators.
टीचर्स थोड़ी खाती हैं, बच्चों की खिलाई हुई चीज टीचर्स नहीं खाती हैं, वो गवर्नमेंट स्कूल नहीं है, ये गवर्नमेंट स्कूल नहीं है कि टीचर ने खा लिया बच्चों का
of iron chelators in Parkinson's disease. If you decrease the iron accumulation, you decrease the disease severity. And of course, let me skip over this. And of course, very recently, there's been this huge upsurge of interest and discovery in the field of ferroptosis. ferroptosis is a form of iron-dependent cell death. It can be prevented by antioxidants like GPX, glutathione, glutathione peroxidase 4, and so on. And it's being discovered as being increasingly in the cell death in many diseases. So the role of iron chelators in preventing ferroptosis may in fact be very important. Many years ago, John Gutteridge and Tony Siegel and myself studied lactoferrin, present in many body fluids and also in milk. When iron binds to lactoferrin, it is rendered redox inactive. And a number of groups have shown that in fact lactoferrin or some peptides derived from lactoferrin might have therapeutic benefit. So my last few minutes, I'm going to switch to another compound, ergothioneine. Ergothioneine was first identified